Today, our topic is DNA fingerprinting technique in molecular genetics, Unit 2, Chapter 5 in Zoology. So, we have discussed the human genome project. In that, we have got so many results. From that result, 99.9% of genes are common in all individuals. So except the 1% is different, is unique from each individual. So this was found out by, that is the DNA fingerprinting was found out by Jeffrey in 1985, and he is the award recipient of Royal Society Copley Medal in 2014. And we all know that human chromosomes are 23 pairs and it contains 1.5 million pairs of genes. And while studying the translation and transcription process in proteins, not all the DNA is coding for proteins. Only few are coding and other few are non-coding. So they have coding and non-coding sequence in the DNA. The non-coding sequence do their specific functions, that is the regular functions, and the coding sequence code the genes for the protein synthesis. So DNA fingerprinting is useful for identifying differences in some specific regions in the DNA sequence. So this DNA fingerprinting, the DNA sequence is doing the specific function. For this specific function, it has to repeat. The DNA sequence will be repeat in some cases. That DNA is called repetitive DNA. So for some reason it is got repeated. The sequence is got repeated. So DNA sequences is, that is adenine, guanine, cytosine, thymine, and again thymine, that is adenine, guanine, cytosine, thymine. So like this sequences will be repeated again and again. So that sequence containing DNA, it is called as repetitive DNA. So and this repetitive DNA, that is how we will, the repetitive DNA, the bulk DNA, what I had seen in the human genome project, we have get 99.9% is same in all the individuals, in all the people. Only 0.1% will be unique, that is will be different. So how you going to separate this? With the help of density centrifugation. So centrifugation is a machine like our mom is using mixer grinder for separating the sediment and the liquid, making chutney. Like that, here the centrifugation is done for what? To separate the DNA, that is bulk DNA. Okay, the bulk DNA is considered the bulk and minor. So bulk DNA is from 99.9% and the minor DNA is from, that is the small DNA is from 0.1%, and you're going to variate the differences. The small difference only is there, no? So that differences. So while centrifugation, you get a 2 peak. So this major peak is called bulk DNA. While centrifugation, you are getting this. Okay, it is a bulk DNA. And this small, it is called, the small peak is called satellite DNA. Okay, satellite DNA. We can call it as SAT DNA also, but it is satellite DNA. Okay, so now you come across now the DNA fingerprinting. It forms the repeated sequence known as repetitive DNA, and the repetitive DNA is separated that is separated from bulk DNA. So the bulk DNA and the repetitive DNA is got separated with the help of centrifugation, density gradient centrifugation method. There you get the two peaks. This is the major peak and this is the minor peak. So the major peak is called as, that is the major peak contain bulk DNA and the minor peak contains satellite DNA. So depending on the base composition, okay, this satellite DNA is classified into micro and mini satellites. First, what are the base proportion? First, it based on A is to T or G is to T. A is to T is adenine is to thymine rich content or guanine is to cytosine rich content. Based on this, the micro and mini satellites can be classified. And one more thing is the length of the segments, DNA segments, and how many copies of repetitive sequence occurs, that is number of repetitive units. So based on these three, the satellite DNA is classified into microsatellite and mini satellite. Okay, so the mini satellite, we're going to talk about the mini satellite only because we're going to separate the DNA. Okay, so the mini satellite works under, that is VNTRS, that is the repetitive sequence only. The repetitive DNA sequence only we used in the mini satellite. VNTRS means variable number tandem repeats. So variable number tandem repeats, and VNTRS, this DNA is used in as a radioactive probe which going to separate the DNA and forms the complementary DNA also. So it's going to separate the it acting as a radioactive probes. Okay, probes. The mini satellite, it is acting as it is formed from a VNTRS. The mini satellite means VNTRS, variable number tandem repeats, and it is used as a radioactive probes. Okay, so these sequence can be polymerized. Polymerized means can create large number of, that is it has so many shapes. You can create large number of sequences. For what we are doing this? For DNA fingerprinting. To form a DNA fingerprinting, we can polymerize the sequence to make it into large copies to form DNA fingerprinting. So this is an example, and based on this principle only the DNA fingerprinting is doing. So you have this one is a A person and this is a B person. Think like that. So in the first one you can see the bands is 3. In the first chromosome you have 3 bands, the colored bands 3. And in the second, because these are homologous chromosome, no, 2 copies will be there, no, homologous. So first one you have 3, second one you have 4. Same side, this side. And this side, so this side also you can see you can count the bands in the first chromosome and in the second homologous chromosome. And same in the second chromosome you have the numbers. So 1, 2, 3, 4, 5, 7 I think so. And the next one is 6. The same way here also. And in the last one you have 4 and down you have more in number. Okay, same way this side also. So why we are taking that is, this is an A sample and this is a B sample. So for B sample you have to separate, that is A and B samples you have taken, and you have, that is all the homologous chromosome, you have count the bands. The bands is nothing but the DNA bands, that is in the chromosome, the gene. For example, I am saying this. So now you are going to run it in the gel electrophoresis. So that is in a gel electrophoresis, the A and B that you have based on the, here you can mark it as 1, that is starting from the 10 or 15 or 16, you can mark here to the least one. Okay. So and you can keep it, for example the 4 means you can keep it here, you can put the here 4 like that, for example. Like that you can do the A and B samples separately and you can keep it. And you want to know the third person, a criminal fingerprint who had done the crime. So that person DNA, you are taking it and you are doing gel electrophoresis and you are segregating it. And you can see to which person, that is A or B, the C, that is the C is going to match. So it resembles the B person, the C resembles the B person fingerprint, DNA fingerprint. So who is matching? The A is not matching, but the B is matching. So it is due to the copy based on the radioactive copy, that is VNTRS. So different copy numbers it is formed. It is an example. They will find out like this only. The which is mostly used in the criminal, that is to find out the crime, the criminal person. Okay. And they will select the select the samples as hair roots, nail roots, the blood samples and vaginal fluid, semen, like that. From that and all they can able to collect the DNA. And the steps involved in DNA fingerprinting technique is depicted. So the steps or methods. The first step is extraction of the DNA. So the sample of the DNA may be blood, semen, vaginal fluid, hair roots and bones, nails also. And polymerase chain reaction, PCR. So we are taking a minute small amount of DNA. For this to make the small amount into large amount, we need the polymerase chain reaction. Polymerase chain can able to, this reaction can able to multiply the copies more in number at a time. And fragmenting DNA using restriction enzyme. So the DNA molecule is cut into pieces with the help of enzyme, restriction enzymes. Separation of DNA by electrophoresis, that is here. This is the agarose gel electrophoresis and you can see how the DNA molecule, that is the DNA band, the chromosomes are separated here. So that was done with the help of agarose gel. And the DNA fragments are, see, they are separated in bands. No, the DNA fragments are separated in bands. You can see the different colors also here. To know you the differentiation we had given. So you can see the bands also. And denaturing DNA. So the denaturing of DNA is also done in the electrophoresis. For that process, the process heat can be more, temperature can be also denatures the DNA, or alkaline chemicals are also denatures the DNA. Blotting after gel electrophoresis is over. So this material has to be, the DNA bands has to be transferred to a nylon membrane, because to size the fractioned DNA. So for sizing that fractioned DNA stand, you have to change it to the blotting. And here the blotting technique we used is southern blotting. Using probes to identify the specific DNA. So VNTRS, that is the radioactive probes, are used to identify the DNAs, that is which is hybridized. For this they are using the fluorescent substance or radioactive isotopes to prepare the probes. Hybridization with the probes. Now the DNA got hybridized with the probe. And what happened? We have to wash up. So many probes will be there. So only few will be hybridized with the prop and other has to be washed out. So it should be washed out. While washing it, you have to keep the DNA on the film, that is photographic film, to found out the DNA hybrids. So you can see on that is it is a membrane like structure which looks like a photographic film on a membrane, and you can see the DNA hybrids. Exposure on the film to make a genetic or DNA fingerprint. So specific bands. So specific bands will be appeared, thin or dark bars. Pattern will be seen, that is the DNA fingerprint or genetic fingerprint. So I will explain with this. You can see this one. The blood sample. They are taking the blood sample. This is the first one is the blood sample. From the blood sample, the DNA is isolated. So the DNA is isolated and cut into fragments with the help of the enzyme restriction endonucleases. And you are allowing for centrifugation, that is to make it, that is you are allowing it on the gel electrophoresis to separate. So gel electrophoresis, after the gel electrophoresis technique, you are separating the DNA, that is AB, chromosome AB, like that you are separating, and you are forming the bands with the help of, that is radioactive props is used for hybridizing the DNA. After the hybridization, that is after the radioactive hybridization, everything was over. The DNA is, that is the DNA fingerprints are kept on a photographic film. That film, that is the excess of a hybridized, is washed off, and you can keeping it on a photographic film which is kept on the membrane. So that membrane, after that you can see a barcoding, thin and thick barcodes will be appeared, and that is called the DNA fingerprinting or genetic fingerprinting, and from that you can find out the for whose it is matching. Okay, so this is the methods involved in DNA fingerprinting or steps involved in DNA fingerprinting. Applications of DNA fingerprinting. Forensic analysis. Forensic means to find out the crime, how the crime and the criminal had involved in this approach, that is in this criminal activities, a person is involved. To find out the person, this DNA fingerprinting is helpful. So forensic field, it plays a very, very important role to find out the criminal. So it's and settling the paternity and maternity disputes also, that is there are some problem, this person is not belongs to me, he is not my child, like that. To find out the paternity means father or maternity means mother. The disputes can be settled with the help of DNA, that is DNA fingerprinting. And determining the relationship for immigration purposes. So the relationship from one place to another place while immigration. So this person will belongs to another country, but he had settled here. So that type of process, that is relationship, can be determined with the help of forensic analysis also. Pedigree analysis means it is easily hereditary disease. To find out the hereditary disease, it can be detected at the womb, in the mother's womb itself. If a mother or a father has an hereditary disease, that is to be there in the child also, that can be detected and rectified with the help of DNA fingerprinting. Conservation of wildlife. So we can protect the endangered species. Endangered means which is going to disappear from the world. So that type of endangered species can be protected, and you can maintain the records of the endangered species, or dead tissues of an organism which appeared previously in the world, which appeared and lived previously in the world, that can be preserved, and now the endangered species tissues can be preserved from the dead endangered species. Anthropological studies. So determine the origin and migration of human population and genetic diversity. Once upon a time, the humans are living in the forest in caves, and they are migrating for the food purpose and their shelter and safety. Once upon a time, that is when they are in nomads condition, the hominids like condition, they are traveling from one places to another places where they get sufficient amount of food and shelter and safety. So that can be studied with the help of DNA fingerprinting. So that type of studies are called as anthropological studies. Anthropology, it studies with the human beings, the human where they had come, where they have evolved, where they had come, and what are the behaviors that we can able to study with the help of this DNA fingerprinting. Thank you for watching the video. If you have any doubts, post your doubts in the comment box. Give a thumbs up, share and subscribe to Science Easy Tech channel.